प्रोटीन आइसोलेशन के बाद जो नेक्स्ट स्टेप है उस प्रोटीन को हम यूज़ कर सकते हैं फॉर फर्दर एनालिसिस, और वी मे नीड टू प्रोसीड फर्दर फॉर concentration of proteins, means to concentrate the proteins. This is also referred as precipitation of proteins. इसकी मेजर वजह क्या है कि जब हम लार्ज स्केल प्रिपरेशन की बात करते हैं, having a large tissue, proceeding further for homogenization to get the tissue lysate, then the volume is quite big. For example, 1 kilogram of the tissue can give you 2 liters of the homogenate. That 2-liter volume is not workable, and at the end of this experiment or this isolation, the working solution will only be 2 milliliter. So we have to adopt some method that all the protein which is present in 2 liters should be concentrated in the relatively smaller volume of 2 milliliter, 1000-fold decrease in the volume. अगर इस चीज़ को सामने रखा जाए तो हम कह सकते हैं कि जो वाटर है इज़ मेजर कंटेमिनेंट ड्यूरिंग प्रोटीन आइसोलेशन. वी मे नॉट नीड सो मच वाटर in the protein isolation method, so we have to remove that particular unwanted volume of water. रूटीन में जो मेथड यूज़ किया जाता है उसे हम कहते हैं अमोनियम सल्फेट प्रेसिपिटेशन. अमोनियम सल्फेट इज़ अ सॉल्ट. इसको जब हम यूज़ करते हैं हाई कंसनट्रेशन में, देन प्रोटीन कैन बी प्रेसिपिटेटेड एंड कैन बी प्योरीफाइड. इस मेथड को एक और नाम दिया गया है सॉल्टिंग आउट मेथड, विच इज़ इन रूटीन यूज इन द लैब फॉर कंसनट्रेटिंग और फॉर प्रेसिपिटेशन ऑफ द प्रोटीन्स. What is the principle behind this? When we add high salt concentration, why proteins precipitate? अगर proteins की structure को देखा जाए, proteins they are having the hydrophobic sites which are covered by the water molecules. A layer of water molecule is made around that particular protein, and when we are going to add high concentrations of this salt, those water molecules they are recruited to dissolve the salt so that the salt makes up the solution. During this process of solubility, salt is going to be dissolved by the water molecule. The hydrophobic sites of the protein, they are exposed, and at some stage they interact with each other. When those sites they interact with each other, then protein starts precipitating. And after a reasonable time, the protein can be seen sitting down or precipitated in the tube or in the beaker as a white mass, which can be separated by centrifugation. इस सारे प्रोसेस में जब हमारे पास इंटरेक्शन के बाद प्रोटीन प्रेसिपिटेट करती है, then we have maximum protein in the form of precipitate, and the upper aqueous phase contains this high salt concentration of ammonium sulfate and the water, which we can easily remove from the separation. Schematic representation अगर की जाए, we add ammonium sulfate salt that is going to cause the precipitation of protein. Those proteins are separated by centrifugation, and those precipitate are then redissolved in the required volume of water or buffer, and if required, the extra concentration of ammonium and sulfate will be removed by an additional step which is called as dialysis.